All right, so let's actually, I kept talking about data, is what the data says, so let's now try to take you through some of the data. This was the part, I was trying to set up a hypothesis, so this is the part where we're going to talk about, evidence that maybe what I showed, thank you, Tommy, on the last slide that we discussed a lot. What's the evidence that ventral stream populations have solved the problem as I've defined it? And I'll show you the evidence we have for this, and I'll talk a little bit about causal evidence that we have more recently. So again, this was to motivate. Here was the idea that things look something like this. So what's the evidence for that? So this is actually work, as I already sort of briefly alluded to, that began with Gabrielle Freiman and Fine Koji were both here in the room, and done by a postdoc working in my lab, Chao Hung. This is way back in 2005, but we just did something very, very simple that I think was asked, why has this been done before? And I was actually surprised it hadn't been done before either, but if it hadn't, we did it. Just take a broad set of objects. These are simple images. Look at blank background. They're very, very simple. There's just like a proof of principle idea. We did a little bit of, we presented those objects at just different scales and positions. And then we went and recorded to a passively fixating monkey. We showed each one for 100 milliseconds. Again, these are these sort of snapshot durations that I already introduced you to. As I said, 100, 200, very similar behavior, but that was what we did, 100. This is randomly shown, just like that RCP video, so you can go measure a bunch of data. Here, this is an old movie I have from Chow. You can listen to some IT here on it, but you might not be able to hear this, but... So, you guys kind of hear that? You hear just some static, those are spikes. The monkey's fixating the dot, that's what he sees, a bunch of images come up, this is a little grainy, but you get the idea. And you stop, so you go and record an IT, and that was sort of one neuron listening to a single electrode recording. Here's just this is probably the first neural data of IT I've shown you in detail, just to give you a feel. From my lab, these are these images, these little tick marks, those are action potentials there for those images, and they're randomly interleaved, and then you present them many times, you get randomly interleaved, and you can see, hey, this site, this neuron IT likes this image, and likes this one, see this response, latency of about 100 milliseconds. Doesn't like these as much. You can see variability in response. Here's what real data look like. Here's another site like this, doesn't like that a little more, needs a little less. And then here's another site in IT, okay, it really likes this for some reason, and this as well, but not so much those. Okay, so that's a feel for the data. This is what raw data, spiky data IT look like. Chao and Gabriel and Tommy and I, we collected like 350 sites at the time. These are shown here. These are now the mean responses over those large set of images, or not very large by today's standards, but at that time it was 78 images. These are the mean responses. You shouldn't read anything from this except we had a lot of data that you could construct populations for. And remember, the goal is to just test how good is this for doing the task, being accessible, linearly separable, separating say, object A from not object A. I'll show you those categories in a minute, versus something that was a little more inaccessible or tangled. Here's like this sort of XOR layout here, that this is not easily accessible. So good, bad. Simple idea, ask how well you can do this. And, this is a slide that just shows if you had a simulated, if you had a population activity that emerged from response to one image that we measured. We did things like count up things, numeric spikes in one interval. We could vary that interval, but just think of the spike counts in a 100 millisecond window. That gives you one number. So you have now, if you had n neurons, you have n numbers. So and we did that for a whole, we asked how well could you from this predict which object category was there. We had 8 predefined categories. I'll show you the names in a minute. But, oh, there they are. But really, you can think of that vector as being one point in an n-dimensional state space of IT population. And again, ask how well can you separate a classifier, linear decoder, linear width threshold, as we were discussing, these images from those images. And this would be for instance, space detection. But we could do this for all 8 of these categories. This is just, again, from Gabrielle Chao and Tommy and I's paper a long time ago. It just shows you don't need that many sites to get up to near perfect real categorization performance on single trials on that neat way. You can also do identification quite well. The interesting part to us is that you could train classifiers that say the single object, the center position, certain size, and then test it on different repeated images of that. And of course, you can do quite well at that. That's maybe not that surprising, but what's interesting is you can also do very, very well if you change the position and size, and you see just a little bit of drop in performance here. And that really is reflecting those IT single unit properties here assessed at the population that I showed you about earlier. So there's power in the IT population. This is not, we showed you couldn't easily do this. Later we showed it B4 is not nearly as good as IT at doing this. The input to IT and B1 is certainly not as good. Consistent, as I showed, with a bunch of work. So this was the kind of evidence that got us thinking about the way that I showed you on the last slide. This is how we think about the state space of IT. Now, again, I want to point out that all I've done so far is leave information and neural activity for doing those kind of tasks that could be decoded. But remember, the big picture that I presented at the beginning is we want to be able to map between neural activity and human judgments of these things. So that would be a stricter decoding algorithm, not just saying there's information here about these things, but how well does it predict what the humans are going to report or what the monkey's going to report. So a mapping between this domain, let's say spiking patterns in the ventral stream, or say IT more specifically, to a report in this domain. Oh, it was a car, it was a fish, it was a dog, it was a cat, it was a clock. Okay, so that's called, again, a decoding algorithm. The goal is to build a real model that could be falsified, has testable predictions, and that's what we try to do. So, again, the goal is, here's behavioral report now, not just saying you can get information with decoders out of here, but say, what's the mapping between this and behavior? So that's here. What's this mapping, what's the decoding of it? So these are the folks that did the work I'll show you next. This is Najib Majal, was a postdoc in the lab, Alpine grad student, Ian Solomon, an undergrad at the time. And again, we're in the domain of core object recognition, and what we wanted to do, remember, we're trying to ask, does the neural activity predict behavior? You have to actually then go and measure behavior. So I showed you a little bit of that earlier with the monkey, but now I'm going to show you what we measured. We didn't characterize the whole domain of core recognition. That's challenging, although we made some recent progress on that. What we did do is just try to test behavioral performance on a sample of spanning tasks. And I'll show you those in a moment. You can decide how spanning they are, but I'll just show you what we did. We did this not by just taking images off the internet. We wanted to control the latent variables, so we used 3D object models. We could then control their other latent variables. We could then render them and place them on backgrounds. We tended to place them, we did place them on uncorrelated backgrounds, not anti-correlated, just uncorrelated, meaning the background was randomly chosen with respect to the object. That produces sometimes weird-looking images. Here's some examples here. These are semi-weird, heads floating here. But the point, the reason we like this, first of all, this is a generative space of images, and because of this strategy, we had been doing computer vision modeling at the time. Computer vision modeling at the time were really struggling with these kind of approaches because a lot of the performance was being driven by leaning on backgrounds and tricks like that. So this was something that humans still did really well, yet machines were having a hard time with. So that was a big part of the reason that we chose this kind of approach, to try to isolate where humans beating current machines at the time. And, to give you a sense that you can do this, there you go, okay, car, plane, base, car. Okay, those are a little grainy, but I think if I can cue you, you could notice that, again, those are about 150 milliseconds, and you should have been able, you probably could do almost every one of those. Maybe you couldn't tell because I was saying the word, but you could get a sense that you could do that quite well. So it's the same thing I showed you earlier now, just a more defined task space. So I mentioned we're going to sample a bunch of tasks, expand it. We did something that would be referred to as basic level categorization first, so we had 8 categories here. And there were 8 objects per category, and we would generate these images at low, medium, and high variation. What that means is those other latent parameters, we would have them restricted to low range, medium range, or a high range. And I'll give you a sense of that kind of images here. So here's faces generated under high variation, so lots of changes in position and scale. You can see the random backgrounds behind them. Here's some not face objects, again, generated at high variation. These are just, think of these as distractors. So we like to just break everything down to a binary discrimination. This is discriminating face from non-face. So what I'm going to show you is a lot of binary discriminations, performance estimates that we made from both humans and neurons. So the simplest thing about this binary discrimination. Again, here's a linear classifier. Can I separate that from that?
Can you say again why you want to use these
the crazy backgrounds?
Strange, yeah.
Instead of
realistic backgrounds?
So the main reason is efficiency. So if you could eventually show that you could bring machines worse than humans with enough images. But you don't. But you need a lot of images to show that often, and we can't test that many images in the neurons. We wanted something that would isolate things. It's still be challenging, but it's more compact. So that was one reason. The other is those other latent variables are also very interesting. As I mentioned earlier, we think IP makes it contain that information. But on random images pulled down from Google or ImageNet, you can rely on human annotation, but you don't actually have access to ground truth in those things. So that was the other reason.
You could generate images just in consistent context.
Consistent context, right. We were trying to isolate objects as opposed to what you might call context. And you might say, well, okay, I'm interested in context with interaction. Obviously, we are too. We just wanted to start with a clean single object case. So this was our attempt to do that.
There's still an interaction here with with contact.
No, there's no interact, there's no,
Well, when I see the plane hitting the ground, I immediately am very surprised by this. And it might be a different sort of mechanism that I'm using to identify it than if I were just to see it flying through the air.
right, so I guess we're just asking you to identify, give me a name, and you saw an image, and your job was to say plane, and you might say, well, I'm not doing the right, I'm not using my normal plane mechanisms when I do that. I guess we could criticize that, but we would assume you are. I don't know.
I would guess anything context would hurt there because you don't expect to see a plane hurt the ground.
Yeah, but I mean, it's quite surprising.
Once used for a while, it's not surprising. You get used to like, okay, the background is kind of like black, and that's kind of what it is. The computer vision though is like, oh, I can't cheat anymore, and the line is felt to zero under these conditions. So that was really, and it was easy to show they were, a lot of them couldn't do anything with these kind of tasks, for sure. I'm not trying to claim that this is all of vision, or all of it before recognition. 2001.0, I should have mentioned that. This was our first step. I'd rather motivate the strategy of generating images, test behavior, test neurons. And I'm going to show you what we've done so far. So all your criticisms are well taken. This is not the end. It was just what we've done first. I'm sort of giving you a history of how we got there. Okay. So keep that in mind as you show you the results. So we didn't just do basic, but we did car identification, so subordinate tasks discriminating, I don't even know what these are, Z3s from Beetles, different cars that are slightly different from each other. Here's faces. These are hard to tell, but these are actually different faces. They don't have hair or glasses or other cues, but they're actually different shapes. This is hard face subordinate identification test. I won't take you through the behavioral studies. Humans were basically doing 8-way tasks, and then from that we could sort out their binary or two-way discrimination from those data. I won't take you through how we do that, but think of each of these as a binary task. So this is discriminating. That right there is animals from all of these other categories at low variation. And this is the D prime level. So high, red means very good performance. 5 is like nearly perfect. The D prime is infinite at 100%, but nearly perfect. And 0 is chance. So you can think of that's about 50% correct on a binary discrimination. So these are psychophysical terms that capture things like biases and how we measure D prime, but this takes us a performance measure. So you can just see, what I want you to see is that, hey, subordinate's harder than basic. That's kind of reflected in the colors. That's sort of obvious. Some tasks are harder than others, or for some reason. And the high variation is harder than low variation. Sort of all this is intuitive. You might say, well, this is sort of obvious from the physics of the world that this should be true. But you can't take low-level pixel models that easily produce this behavior, and I'll show you that in a moment. This is first of all, humans find some tasks harder than others reliably. That is, these are not noise in the data that I'm showing you. If I took 2 different humans and plotted it here, these are pooled human data, but if I show you different humans, they show the same pattern, and I'll quantify that later. So the pattern's reliable across humans, and again, it's not explained by low-level features. Even though your intuition tells you this is what it should be, I would argue using intuitive psychophysics to do that. This is constraint data for us. These are the kind of data that should be predicted by a decoding algorithm. If it's correct, it should show these same patterns that we see here. That was the first level thing we would like a decoding model to do if it was correct. Not the only thing, but the first one. So we play the same game. You present an image. We measure a bunch of neural activity. We can count spikes in a window. We just sort of simulated data here just to show you. To measure the response of the neuron, you get n neurons, you get a point in n-dimensional space. That's to that image. Now we have these complex images here, it's the same game, got a lot of images, you want to say, how about kind of separate these labeled green face images from non-faces? Again, this is a hypothesis now, not just is there information about faces, but a hypothesis is that this kind of mechanism supports face detection behavior or reports. That's the hypothesis. The prediction is that the algorithm, as implemented here on this neural basis, should predict the human/monkey behavioral performance on face detection tasks, and it should predict this for all tasks that we measure. So it's not just predicting for one task, but a whole battery of tasks that I showed you. That's the prediction. If you believe that the model that many did like, that you just take a linear decoder and you do this, it should predict the performance. That's the prediction from that. So how do you test it? We go and measure a bunch of neurons first, just like we did before. In this case, we're not doing it single site at a time. We're planning recording arrays along the ventral stream. Each can record from 100 sites in parallel. So we ended with samples of a few hundred neurons again along the ventral stream, IT, various parts of IT, and also V4, the dominant input IT. So we can sample among several monkeys. We pool the data together to show you what I'm going to show you next. And now here's a response vector. Those greens indicate the mean response of 168 IT neurons to that particular image. Here's a bunch of other images. And we actually collected now thousands of images, not 78, 25, 60 images over lots of variation that I alluded to earlier. This is just one data set out of IT for a bunch of, so this is just the top 30,000-foot view of the IT mean response data. Of course, we have the spikes and everything trial by trial. And remember, the idea is it doesn't predict performance on all the recognition tasks. When I say this, I mean this. We need to predict D prime values for all the 24 tasks we saw on the bottom of that previous slide at all the levels of variation. We had 2 to 3 levels of variation that we tested in there. And when I say does this predict, I meant this hypothesis. We have a linear decoder weighted on this, appropriately weighted on this to predict performance. So again, we think of that as being a downstream neuron with some appropriate weights, and this is the signal to say, oh, look, I see car. That's the biologically plausible hypothesis to us. So I'll just show you the key result. The key result is that when you do this kind of decoding algorithms, you apply the same decoding algorithm on the IT feature space, which I'll call the code, that it accurately predicts human, and we think monkey, although this is tested in human behavior, and as I showed you, humans and monkeys look similar. We haven't done monkeys in exactly these same images, but it at least so far predicts human behavior. And the algorithm in words is basically this. Sample roughly 150 random sites spatially distributed over IT. This is similar to what I implied before, but I didn't lay out in words. So they're random, they're distributed. We measure each site's average response over 100 milliseconds. Again, that's something we can vary, but what we did was 100 millisecond averages for each object, learn an appropriate weighted sum. We can talk about the learning algorithm, basically the type of classifier, how many training examples, I'll show you that, but it's the same for all tasks. And then that, to me, is a big complicated algorithm that's learned weighted sum of randomly selected average 100 millisecond response distributed over IT. That's what we were implementing as a hypothesis of what's going on, a decoding algorithm. That's still too long for me, so I shortened that with an acronym, laws of RAD. For better or worse, that's what I'm going to call all of this going forward. Okay? So it's just an algorithm that has a name. It's not just a hypothesis. It's a specific algorithm. Here's part of the evidence that I sort of alluded to that we think this is really impressive to us, that all we did was apply this algorithm. Here's the predicted performance of the algorithm. Here's the actual performance from the D prime plot. So D prime 0 to 6. You know, here's spaces versus all the other objects. Remember, these are binary discriminations. And what you see is that this is almost on the unity line here. So 64 object tests. And these, you might say, well, this isn't perfect. It's almost on the unity line. And the spread off of here is no greater than the spread. I mentioned that humans were very similar, but if I put a single human here and the pool of humans here, which is what's on here, this is the pool of mean human D prime, and if I put a single human here, that is indistinguishable from this here. I'll quantify that for you in the next slide. Here's the human-human consistency. This is now correlation values. So take the last slide, this is just the correlation. So it's really how much does this pattern, forget about the unity slope, but just does the pattern, does it lie along a line here? Is it highly correlated? Ignore the performance here, which is important. But for now, it's being ignored. That's just the pattern. Here's that hypothesis. You take a learned weighted sum of IT neurons. You're right up here. We can do a lot of other things in IT, which I'm only just showing you one here. That's the best site we recorded in IT for each task. That's a certain type of decoder, kind of a grandmother cell-like. Here's where it ends up. You do things like you take D4 and do weighted sums on that. You don't get nearly as high. You take D1, you don't get as high. These are things I implied earlier from the earlier study that I mentioned. Take pixels. I mentioned low levels doesn't do this. You're not getting predicted patterns of humans. You can take computer vision algorithms at the time, use some of them here. None of them come close to this. But this algorithm is hiding, sort of passing the Turing test, if you will, decoding off of IT in the way I described for you. To all extents and purposes from these tasks with these measures is indistinguishable from another human being. So I recorded out of IT, applied to that out. So that means it's consistent so far with the data, not just with the correlation, but it's hiding in the range of perfection for the moment. Okay, so that's part of the reason that we think that this algorithm is a viable hypothesis at the moment. We can also look at the confusion patterns, not just the pattern performance across those tasks, but what kinds of errors are made. So here's ground truth. Here's what the algorithm is reporting, the kind of mistakes that will be made. I don't have the scale for you, but red means more mistakes of a certain type. Blue means less mistakes. I just blanked the diagonal, because we're just going to look at off diagonal. Here's the actual human data measured on those same images. And remember, these aren't single images. These are pools of images, so these are averages of mistakes. And so it's not yet at the image level grading, but you can see that these look very, very similar, the off-diagonal elements. They're not perfect. But noise-corrected correlation is actually 0.91, so it's almost perfect. So 1.0 would be as perfect as we could get within the noise limits of the data. So it's really, really good, even on its confusion prediction, again at the object level. Now, that's median variation. The high variation, for reasons we don't yet know, suggests the algorithm is not perfect. There's things that are wrong, and this is interesting. It's only at 0.68. These colors are, sorry, a little hard to read, but it's only 0.68. You know, somebody could write a paper and say, wow, 0.68, that's really cool. To us, this is a failure. This is a model. This is a deviation from perfection, which means there's something wrong with the model, and we need to understand it. Maybe that means we're looking at the wrong time window, the wrong area. But that's where we can dig in and sort of refine the algorithm going forward.
Sorry, can I ask a question? Low, medium, and high variation, is that variation within a class?
That sort of, think of like you're doing all those tasks, those 24 tasks I showed you, either with little variation, that's low, medium variation, that's the more pose and size of position, and high is
okay, so it's kind of the input, the tweaks
like
of the
how
input.
much of the generative space, how much sort of variation is there in the object projection.
Okay.
And so these are, we kind of ran those as separate groups. This can sometimes contain some of that, right, because we're just expanding the range, but it's a broader sampling of the range. Okay, so I'm going to skip, I mean, there may be those that are in the room. I'll just quickly say, if you guys want to talk about this, you can look at finer rate codes. This is something Gabrielle and Tommy and I did way back when the earlier paper. They might improve things a little bit. This is 100 millisecond average. These are finer rate codes. They might improve a little bit. You see there's a trend here. But all of them are sort of within the levels of our current data, so it doesn't improve things much, and so they're all passing the Turing test. If you look at, because these neurons recorded simultaneously, you could ask about correlated data versus not correlated data. At least for these tasks, there's not much difference there. If anything, it seems to get worse if you, there's a trend slightly better, but it's not much different. And you can also start getting into the face literature that I talked about earlier. If you think that the faces are supported by the face blobs, you could build decoders that only work on neurons from the face blobs. That's something we did here. I won't take you through it other than to say when you do that, your predictions only get worse. So in our hands, that doesn't seem to help much, assuming a prior like that does not help predict the patterns of human behavior. So remember, faces are one of the tasks in here, and face discrimination is one of the tasks. You just sample over IT and learn, and then you do better than if you start to do things like, oh, all the faces must be done on the face patches. So it doesn't make things better to do that.
Were you recording for the face patches?
Yes, we're recording over most of IT solo samples. Some of the records happen to be near parts that cover some of the face patches. Now, we don't have MR in those animals, but there's so much MR, beautiful stuff from Doris Howe's lab that shows like where it is our own lab, that they show you has gone in and measured those same things. So you almost don't need MR to say, oh, this is the one, this is likely the middle face patch. And so that's what we did for what I'm saying that. And then you can measure the neurons in terms of their facial activity and so they're comparable to those measures as well. So other hands here.
Are you always testing a validation set?
I'm sorry, I should have mentioned that. All the predictions are, you train on some images and you're always testing on new images. I should have said that early on. So everything is predictions on new images. When I say that predicted performance, those prediction performance. Is that your question? Yeah. For every time I show you a decoder or a classifier or something, I've always done that. Sorry, that was true. But I should have said it. Yeah.
The higher variations that didn't go as well, couldn't it just be that you're not looking at enough dimensions, enough neurons?
That's right. That's the kind of stuff we want to dig into next. So you can, so what we want to do now is start looking at, well, as we plot, I'll show you plots as a function of the number of neurons, but it should show up in those kind of plots. And if I haven't done that yet, I just because my grad students are busy with six other things that I'm not telling you about. But that is exactly the kind of direct expectation. We just don't have enough sampling to make those predictions.
You do see a difference in, like...
You get better as you add more neurons to a point. I'll show you that. Again, I can show you that not for the off-diagonal, the confusion matrix, but I'll show you that for the performance pattern, which was the first diagram I showed. Remember, the performance pattern is like the diagonal of the confusion matrix, but just collapsing all the off-diagonal elements. That's the coarsest grain prediction. The next finest grain is the confusion matrix. The next finest grain would be an image-by-image level prediction. And then the next finest grain would be an image-by-image, trial-by-trial level prediction. Right? So we haven't done, likely on all those grains, but we started with the coarsest grain.
You thought medium and high. Is low even better than 91?
It's comparable. I mean, it's also very good, but a lot of the algorithms can predict well at a low variation. So like V1 can predict pretty well at low variation. What separates out those other algorithms actually is going through those harder tasks. So here I wanted to give for neuroscience and remote mechanism. We talked a lot about this. Like what is this mechanism? Well, what we think about is there's some patches in IT. Remember that millimeter scale stuff I talked about earlier? This is just how I think about it. This is a hypothesis. You know, if we take millimeter scale regions, you take the size of IT, that means in central and anterior IT, there's about 150 millimeters squared of tissue there in each hemisphere. And remember, each neuron is doing similar but not exact things. If you imagine that you've got a downstream neuron, this is a simple model, but it's a model, that samples from 50,000 random neurons distributed across it, has 100 training exemplars, say, to learn cars. At the end, it ends up with 5,000 heavily weighted neurons, at least that's what our classifiers look like. About 10% of these end up being heavily weighted. Remember, we're not doing 50,000, we're inferring this. We're doing much lower numbers, on the order of 150, as I showed you in the data. We can infer this, I'll show you that in a moment. But we then end up with about 10%. You're going to have neurons, you can imagine neurons that look like this. You weight roughly 5,000 of IT, and of course you're going to weight the features that are most useful for discriminating the task based on the training exam. One of the things I want to point out, this is to connect neuroscientists to the idea that this is not just black box decoders, this is what downstream neurons could do. And one of the things I want to connect is for faces, for instance. These red blobs are meant to be those space patches I showed you earlier. Here's a neuron, starts out, it doesn't know what to do, but it learns about faces, 100 training examples, 50,000. It would end up, almost by definition, leaning a lot on these spots, because that's how they're defined. They're defined as good at discriminating faces from non-faces. These are good features for discriminating faces. So you would end up with something that might look a lot like this at the end of the day. Still 5,000 neurons, we think, still 100 training examples. The point I'm making on these slides is this is the same algorithm producing different decode outputs, but it's the exact same algorithm that's producing everything that's shown you so far. So one learning strategy applied to an IT basis gives you the data that I've been showing you so far. And it includes, if you like the idea that face neurons support face tasks, it's not at odds with that, it just encompasses it in a larger framework.
I think that's doing pretty much what you're doing a schematic of on the bottom there. It makes face detection worse.
I'm sorry?
Because I thought you made a point about just including neurons in the face patches.
Oh, so this would be a case where they're randomly selected. This is our preferred model. You could do a version where you limit this downstream neuron to only having lines from the red patches and then learning. And that's the slide I briefly breeze through. And then, yes, you'll get a neuron that's still taking some degree of face detection, but the performance predictions across a bunch of face tests don't correlate quite as well with the humans. Now, it's a trend. It's not significantly worse, but it's not better.
And do you have any subordinate kind of face tests for that? Because you can imagine that.
Well, there are subordinate tests. I mentioned we do subordinate, and we did test them, and I'm going through it quickly. And yeah, if you want to go back to that, I could show you on that slide. There were subordinate tests. I mean, the hypothesis about face neuron supporting face tests is not well defined, right? It's supporting detection, subordinate discrimination, everything with faces, bug faces count, it's just not well defined. But in our hands, it was discriminating human faces from each other and detecting human faces from other categories. And we don't see that you need to bias the downstream learner yet to make the predictions fit our behavioral data better yet. But we don't see evidence against that idea. So we prefer a parsimonious idea that so far captures the data. And at its end state, ends up with something that I think people who like to think about face patches shouldn't be unhappy with, because this is sort of what they might think. All right, it's really ultimately a question of learning here to start to distinguish this. Now, maybe when we do image level analysis and we push harder, we may find that predefining a prior, put the neurons to read from the face patches might lead us to slightly better predictions when we push harder to the question here about why is it not perfect. And that's something we'll have to explore going forward. So I'm telling you kind of where we are so far in contextualizing with respect to face patches, which I introduced earlier, so you guys understand.
So, according to this model, how flexible are those downstream neurons? Are you making this claim that we've eventually got somewhere car neurons and face neurons, or are these the connections more defined by task, how you're reading out defined data?
Yeah, so again, this is something that we hypothesized these neurons could live in downstream areas like prefrontal, parietal, and amygdala. And if you think about it strongly, like maybe these are now the grandmother cells in this version, right? Somewhere you have to, whether these could be dynamically engaged in prefrontal, depending on task conditions, somewhere you kind of have to believe in at least dynamic grandmother cells, but to the extent that we can do the task, you've got to have some cells somewhere, that's how we're computing. And we haven't found them, but Earl Miller's lab has, again, trained monkeys do categorization tests, and they find category like neurons in prefrontal. He and Tommy and Dave Friedman published that a number of years ago. So we like that idea, but we haven't gone after these neurons directly, but we think it's consistent we sort of are wanting to put what we've found in the context of what other people have shown, and this is how I think about it, but you might go in and say, well, go find those neurons and now go record from them. And again, some people have gone, but we haven't done these apps yet. So I don't know if I answered your question, but it's a hypothesis of what might be there based on the decoding that we're seeing on the mic. I mean, that's not really satisfying to anybody.
Yeah.
But that's where we are. The part that we like is that you can just take the same algorithm and you can predict a lot of stuff. You don't have to do anything fancy there. So that's the part we really like. So I mentioned the number of neurons thing. Here's somebody asked about that, the increased number of IT neurons in decoding. This is the consistency, that correlation. Here's the Turing area in the gray zones. Here's the red identified there. And when you're in here, you're passing as a human. This is the performance. That's the unity. Like this is what you would just do, high performance in a computer vision sense. In general, if you add more neurons, you always get better, but you don't increase the consistency, the correlation with humans. You don't kind of find the same patterns of difficulty that you. So that's why I've been stressing that as the lever that's more interesting to figure out what human brains are actually doing. If you increase the number of neurons, you of course will kind of get better and better and better, but that's sort of not as much of a constraint. But it allows us to make an estimate that I've already alluded to. You get to that point, it's around 500 neural features with this particular decoding strategy. And I want to point out, though, I sort of mentioned this before, that this depends on the number of training examples. So this is with 100 training examples per object. This is a manifold of possible solutions that would all give us equally good predictions right now. So with 100 training examples per object, you end up with the predictions I showed you. That means you need about 500 neural features, as I just implied in the last slide. Those features are actually computed by averaging data over trials when we do the analysis. So to back this out to real neurons, you have to make some adjustments for real neurons and their noise levels, and that ends you up with a number that's more like 50,000 single units. So that's actually where that number comes from. So this is kind of algorithmic level of what we did to the data, but this is our inference of what you need to support the real-time performance. So this, again, you could pick a different point depending on how much training you think something gets on these kind of categories. We don't know. What I showed you could just pick one point here and you could do all the tasks that I laid out.
I didn't understand what's the relationship to the red and black.
Okay, you get this part, you get this, right? This is training examples, this is number of features needed, and this is the family of solutions that all make the same kind of predictions that I showed you. They're indistinguishable by us. Okay? You got that so far?
Yeah.
Then you want to know about the mapping between the red and the black. That has to do with the fact that when we did these analyses, we first averaged the neurons over many, so there's two parts, but let me make the simple one to understand. You present an image 50 times, same image. You just average those responses together to get the response of the feature, we'll call it, to that image. The brain can't average over 50 presentations, it has to respond to one.
Okay.
So we think, given the noise level, so we can then estimate that everything still seems to work, but we can't measure 50,000 units for all these tasks. We can't do that. But we
100 is just for averaging the...
It's mostly for averaging neurons. The other part is single unit versus multi. So these are multi-unit averages often. Step off the single units, we throw it out as data. That changes things a little bit by about a factor of 2. So this is just a factor of 2. Again, those are inferences from a sub-analysis that I didn't show you here.
And this is for a specific number of categories.
This is for predicting those 64 behavioral tasks, which are those 24 categories over different levels of variation. Remember that funny D prime plot with all the colors? That's her ability to do that, to predict that. That's what it is. Okay, that's the number of output neurons on IT. Some people think this is too low, some people think it's too high. I don't know. I mean, you see there's a 3-parameter here. How much training examples human subjects get to learn this kind of task? We can start to begin to study that in the monkeys, but we don't actually know how to pin that number down yet. So we're describing what data are sufficient to explain. Okay, I want to sort of just say, what is this good for? When you've been building this algorithm here, it's a predictive algorithm. Well, now in principle, we should be able to predict, if it's right, we should predict for any task in this domain. Result, you should be able to predict the pattern, the behavioral performance, and we should be able to predict if we manipulate IT neurons. This algorithm helps us to know under some assumptions what should happen to the performance. I mean, if I can go in and push the neurons around, you make predictions about what should happen behaviorally. I think that should be sort of obvious to you guys. And they're more specific than saying, I push face neurons and face task change. It says I push this neuron over here, and these pattern of all these tasks should change it exactly this way. So you can make all those predictions. I have a slide on that if you want. So one of the things we're doing now is we can move neurons around with learning paradigms to change the IT neurons, and the behavior should then change in a very quantitatively predicted way. I don't have time to tell you about that. I'm going to briefly tell you about our optogenetics work, where we're, again, trying to, in this case, silence neurons, and then it produces predictive perceptual change. I'm not going to give you what you want, what I want to, which is to change like one of those features, that wobble it up and down, and then say the behavior should move exactly this way. That's what in principle is possible. What I'm just going to show you is some proof of principle that we can move neurons around and they affect these high-level tasks. You're going to have to wait probably a couple years till I can show you the results of what we're really trying to do, which is let's manipulate these neurons or groups of neurons individually and see if the predictions follow. But does the spirit of that make sense? You guys, if the hypothesis is right, I should be able to go into those 150 little cubes, if you will, and toggle them each up and down, and there should be quantitative predictions across a range of behavioral tasks if this kind of model is correct. That's the way we're proceeding to try to test this more deeply. You see puzzle books, does that make sense? No? Oh, okay. Okay, so back to the slide here. Remember, we can go map things very precisely. So what we wanted to do is, what if we silence just those neurons? You know, you can get into the face question and be like, what are the face tasks? How does it affect clock tasks? That's one way to think about it. But for us, more broadly, what if we silence that? What happens to tasks if I just shut those neurons down briefly? What if I shut down those neurons? What if I shut down those neurons? That was sort of what we wanted to do. Little millimeter chunks, if you will. This is Arash Afraz, a postdoc in lab is doing that. How do you shut down neurons? Well, now there's these magic tools, optogenetics. Everybody heard about optogenetics? Okay. Magic tools, shut down neurons quickly. That's what's cool about optogenetics. So you've never had the ability to do that before. It's not by activating neurons, it's by silencing them briefly. And so here's an example of IT neurons driven with a visual stimulus. You've seen this before. Latency, here's 2 neurons. Turn on the laser light, boom, shut this down. So block that with the laser on, and it comes back right away. Now, these are 2 of our stronger sites. It's not always this good, but this shows the potential of what this technique can do. Okay, so I'll just show you briefly the behavioral effect. This is Arash's data on discriminating males versus female faces. So this is a gender discrimination task. It's not one of the tasks I showed you. Again, this is parallel work in the lab that I now want to try to apply to story I've been telling you. But Arash just wanted to say he would discriminate males and females, train monkeys on a male versus female gender discrimination task. Here's the monkey's performance. Here's the upshot. You interleave, you turn the light on in particular small regions of IT at a moment on certain trials and not others. Interleave randomly, shut down neurons with a laser. You get a 2% drop in performance in the contralateral field. That you get no drop in the ipsilateral field, and that's sort of predicted with these. That's what we would have predicted given the layout of IT. So this is all I wanted to get you excited about, that we can manipulate high-level behavior. You might say 2%, that's really tiny. If you're interested in that, I have a cool slide at the end to talk about why is this so small. We think that's consistent with everything we know and that I've told you so far. But I'll try and I can show you some of that in the end. But it's small but reliable. So
can you say a little bit about the state of optogenetics in non-human primates?
So yes, far below where the mouse is, but the state is you can put viruses in, they express, and then you can get kind of plots that very few people have shown. Nobody's shown anything in IT yet with that, but you can show some behavioral effects in some places, but people certainly can show that Bob Desmond will have and others have shown you can activate or silence neurons with these kind of techniques to some degree, but not as strong as you can with a small pharmacological inactivator. So the state is the tools are doing something to the neurons, but they're still not sort of tied to certain cell types yet, or certain layers. They're not as precise as they are in the mouse. That's the state. But there's we can have a whole talk on it.
Is it like a permanent change to the monkey?
Oh, well, the neurons are infected, and they're infected for a long time. I think, we don't know that fully in monkeys, but at least months. And those neurons are stable. And, this is what all the work on optogenetics, Ed Boyd and Carl Discaroff and others have worked out of how to do this and what kind of things to use to actually get these to express in a safe way and not lock up the neurons too much.
Yeah, do you notice any other have people noticed behavioral changes?
Not that we've noticed, but there may be things that we're not, measuring. Monkeys not feeling over and dying, neurons are, dying on pathology, but there may be subtle changes, we just don't know what to measure. But we can induce changes by turning on light. That's what I should. Let me just get through this.
Isn't it too general that, there's a face area more or less, and you turn it off? It will be worse. Your ignition isn't a bit independent of doing the model.
Yeah, fair point. So you would say, look, I have a model which says face neurons do face tasks. And if you're doing a gender discrimination, you might say, that's a face patch, because it involves faces. That's what's going on, right? So that means that you would say that you'd already predict that if I shut down neurons and face patches, it should affect a face, that result should happen, right? I wouldn't disagree with you, but I would say that you then can't predict for me what would happen if I actually discriminate a clock from an orange. Should it affect that patch? Yes or no? Face neurons, what should it affect it? Don't know, right? It's just a weakly predictive model. It only predicts sort of some domain that we don't actually understand. So again, it's not saying that there's not some predictions, they're just weak predictions. So they just make tighter predictions about other tasks, which I can't yet show you the power of yet because we haven't yet tested. We did the proof of principle, which is consistent with the mental model, which is why we did that, to kind of test the thing that everybody thought would work, to make sure the tools are working. Then we want to do the more interesting thing, which I wish I could tell you, but it's still going to take some time. Right? But one thing that is cool, and maybe is here that maybe I'll just go really quickly through, is that it turns out there's a little bit of decoupling. It's not that the face versus object selectivity as usually defined, and remember that red and blue dot, is the best predictor of the effect of the performance. It's the gender discriminability of male versus female in each bit of 1 millimeter tissue that's the best predictor of performance, and that is not perfectly correlated with the face versus object signal. So it is correlated, but not perfectly correlated. So there might be sites here that like discriminate males versus females, but are not as good at faces versus objects. Others here that are good at faces versus objects, but not as good at males versus females. So there is some decoupling there. It's not super strong, but there is some decoupling there. So I think it's more interesting than just saying the face patches do everything with faces. I think it's much more subtle than that. But that's sort of, again, the edge of the video.
So I'm interested in that then, because how do you reconcile then, like, Doris's lab and our lab has data that if you stimulate face areas, you get these really big face effects, and then not for clocks or objects or anything. And then the decoder would suggest that you can do it outside of the region or inside of it, and it's just as good either way.
No.
So
the decoder doesn't say it's just as good outside. Remember that was the slide that said when you decode, you're going to have some face signal in IP that you're going to read from.
Yeah, you can like, right.
You're not as good.
Classify
If you're restricted outside, you're not as good. Okay. The patterns actually look very similar to the human patterns, but you need more neurons. So you're not as good per neuron. Okay. That's what the decoder says. So sorry, so just to be clear about that. But so sorry, go ahead with it. You're talking about the idea that if you stimulate here, that you're going to see the nice series of patches downstream. Is that the thing that you
or just like the information seems to be like you can classify over like many of these neurons spread throughout IT. But then if I stimulate, I can affect the perception of like just faces in a face patch. So like, even though the information might be present across it, is it really only the stuff inside there that's involved in perception?
I think that's still the edge of the work. I know I saw Doris at SFN, there was a cool poster on stimulation and the effects of that. Are you talking about Midrick's work now? We say you.
Yeah, hers, or we're Girl Spector's labs with the electrode stuff too.
Oh, the Parvici work? Yeah, so I think there's clear evidence that you mess around with these things, you're going to have something to do with faces. I don't think our data argue against that.
Okay.
We're trying to move to a domain where we can start to predict what are we going to do for a larger range of tasks. So that's where the larger model comes in. And then what might end up happening is the idea of these being modules might become a little more muddied in that. We have sort of more of a distributed view of this system, but it kind of depends on the downstream reader ultimately. I don't think there's any odds right now, but I think there are interesting questions there about, for instance, one practical one is, do these neurons do anything but faces? Like, you might predict, if you believe the strong form of this, when I found these neurons, when I test what clocks versus oranges, it should have zero impact. That, to me, is a prediction of a modular phase hypothesis. If it has an impact at all, then you better throw away that strong hypothesis, right? And but,
You already know that the hypothesis isn't true, though, right?
What?
From fMRI data, we already know that that's very strong.
No, that's a causality hypothesis of neurons. We don't know that from fMRI data. We know from fMRI data that there's information about clocks and oranges. The deep hypothesis is that information used by downstream areas, and you can only test that with direct causal interventions. That's the crux of I think that people's debates about models. Nobody's debating that there's some spatial structure in there, as I showed that earlier. That's there. The debate's about how is it being used, and I think that's still open and interesting. And I think it's going to be fun to see how this all plays out. But I think all we're trying to do is set up a domain of stuff that we'd like to consider a lot of tasks. We'd like them all to work out in some consistent way. And whether you're better to think of, let's study faces and let's see objects as if they're two completely separate areas, maybe that's the better way to think about it. But we like to think about it as a unified space for now. Maybe that's less important. I like to wrap my mind around it that way, but it may turn out to be better explained the former way. I think that's people come at it from different priors, if you will. Yes.
I actually thought that Doris's simulation work did show some disruption with like clocks and objects like that, right?
Right, and this is why, I was a little, when Doris showed that, and then I said, well, isn't that suggestive the strong hypothesis is false? She said, well, but now I think the monkey's perceiving the clock as a face, and that's why it's making those mistakes. So then I kind of didn't know what to say. So, I mean, you gotta make again, this is a lack of predictive, the models have to make some predictions, and I would think that I don't know, if you start to have an alternative use like that, it shows a strong prior, but it's got to be something to the basis, right? I mean, the predictions are what has to be effective, and I don't know, so maybe I'll see how that debate shakes out, but again, I don't think there's a strong debate yet, but I think these are the interesting questions that people are, Doris's lab and Binder's lab and my lab are all having some say on, or hoping to. I'm sorry, let me just jump to the end. I want to kind of skip, I kind of already gave you guys, I told you this in words, but, if you measure the performance, I already mentioned this, you measure the performance of how good each of these regions is to support a task like gender, and then you plot the optical effect of how much silencing affects the animal's behavior. Remember, a small effect, around 2% change, you see that the more useful the features are, the more the effect. It's not perfect, but you can see there's this trend here that the more useful each millimeter is, by thinking of it from a downstream algorithm point of view, the more optical effect you get, which again is kind of proof of principle, but does not engage the debate or the discussion we've been having here. Again, this is just the beginning of these tasks that I said many times. I just want to show you the idea out there for those of you interested in this, that the tools are allowing us and others to start to do this. Okay, I want to end by talking about encoding models. We only have 15 minutes left. But I want to say, I've talked a lot of time about decoding models, and I think that's a fun discussion, the link between neurons and behavior. But one of the things that these kind of models give us is they start to tell us, remember, there's lots of spikes in IP. They tell us, look, if you can just predict the mean rate within this window, then this already telling us so far that's a pretty good predictor of behavior. So they focus on the kind of thing that you might want to need to predict. And so that's the kind of things we're going to test. How well does encoding models predict that? And that's one way that those models help inform the encoding work, but they don't tell you what the encoding model is. They just tell you the sufficiency so far, what needs to be predicted with respect to that problem domain. So let's talk about encoding models in the last 15 minutes here. So remember, this is decoding. Encoding is images to neurons. Okay, that's encoding. Large class of bioinspired models, Jan LeCun, Tommy's lab, Fukushima, all names down here. A lot of people work with this very big class of models. They basically consist of stacked structures of linear filters, followed by nonlinearities, like thresholds and saturations, followed by some sort of pooling operation, usually including some sort of normalization operation, sometimes in different orders. But these are the basic operations that many of these models share. They are all, so these are meant to be models of possible mental scheme algorithms. You might say, oh, this is too forward, or this is too back. I wouldn't disagree with you. I'm just telling you the class of models that have been tried, and I'll show you what we've done. They have the neurons, they all have large span-in like neurons, they have no nonlinearities, they're convolutional models, that is, whatever filters you take, you apply them across the whole visual field. Each layer has many types of tuning functions, so here's like a V1 model. You can imagine different orientation filters applied across the visual field. Here's some other complex nonlinear functions now on the input, now applied across the visual field. That's what convolutional means. And it's deep, and so Jan would say, well, as long as it's level 3 levels, he's allowed to call it deep. Deep means greater than 3, at least according to Jan. So I'm going to tell you about our modeling efforts in this domain, but what I want to do is just give you some context to say, remember, what's cool about these models is not the arguments about the specifics. What's cool is that different than psychology, weakly predictive models, these are models that make predictions that can be tested. So each model, whether it's right or wrong, makes predictions about what layer. That allows tests, which then allow models to be refined, and I think that's sort of the spirit, again, of the CBM enterprise, that as long as you make a prediction, that's better than making no prediction in my mind. A wrong prediction is still better than a model that can just predict anything. It's not useful. So, okay, I want to also say that this large class of models has thousands of unknown parameters, which I'm pulling up in these betas here. These are hiding many, many parameters, as you might imagine, that are hidden up in this class. They have to do with like what the filter shapes are at each level, what are the nonlinearities, how do you threshold, how do you pool, what's the normalization strength. You can imagine a whole bunch of parameters there. And so what we wanted to do is to ask which of these algorithms, I would say if any, many of you already would say this is impossible, they don't have any feedback, it's not going to work, but we said let's just look at the class and ask which of these algorithms, if any, is the one that's interesting. And what we chose to do was to try to optimize for something. Let's optimize to find specific algorithms in this class. What are we going to optimize for? Which algorithms do we want to pull out of this large family of possible algorithms? That is, what parameters do we want to choose? What optimization target are we going to choose? Well, we chose an optimization target, a visual task that I've already implied that we think the vector stream is built to solve, either built by evolution and or development. That optimization target is invariant core recognition task. That's what we picked up an optimization target. So I think when I say we, I mean Dan Yain as a postdoc and I'm a graduate student. And what they did was to optimize for these kind of tasks a large variety of objects. In this case, it was 36 objects, rendered with high variations under the same strategy. They're just on these uncorrelated backgrounds. These are not the objects I'm going to use in later testing. So they were just different 3D objects and we're going to use completely different objects when we do the testing. So we're just making a space of stuff generated from objects that you have to discriminate one for another over high variation. That's the intuition of what we're optimizing for. And so what optimize means is find a model that is a fixed set of parameters, which we call HMO1, because it stands for hierarchical modular optimization, a term they coined for the modeling. I don't have time to tell you about it, but it's published now if you're interested. But you're going to choose a specific set of parameters that is a specific model, not a family of models, a specific model that has all its parameters fixed. And it has 4 layers, which we think of as modeling these areas of the brain, the hypothesis. And again, the model parameters are fixed just by optimizing performance on those kind of tasks. And so once we pull down a model like that, optimize performance on tasks, remember, never looking at any neural data at all, just try to do these kind of tasks well, then what we've been doing is asking, well, of course we can measure all this, let's just take the model features and ask how well they predict the single neuron IQ responses. Is everybody with me so far, what I did? I know you don't know the details of the optimization, but there's a large family of largely feed-forward models, find parameters in there that do perform well on the task, and then all we're going to ask is how well does it predict that. Okay, so hypothesis test. A test of a large space of hypothesis, which we found one under a meta-hypothesis of optimization for performance on tasks. Everybody, yeah, I'm only go.
So, is the idea that, so you're matching the IQ responses, but is the idea that the parameters are learned in a similar way that the brain might learn the parameters?
Yeah, so great question. So we're agnostic as to how the brain optimized to get to this point. It could be evolution, it could be development, it could be both. All we were trying to say with this work is if we just try to find a model in this biologically constrained space that we like, which is the classical model, if we find one that does things that we think IT does, does that offer us any more powerful prediction on the response of neurons than other models that have been existing? It's agnostic to whether that's evolution, we recapitulate evolution or we recapitulate development. Our techniques for optimization are certainly not what we think the brain used in either of those. So just think of it as a way to fish out something of a class and then ask, did the optimization target and the model structure lead to better predictions of IQ? But your question is great because that's really the future. To the extent it does, we'd like to make it learn or evolve like the brain really did, which is much harder, but also almost more interesting in a way. But this is to try to get us in the game. Yes?
So is the prediction based purely on firing patterns in IT?
So you're asking, what are we going to test? So this is a question. I haven't shown you the result yet, but we're going to ask, how well does this predict that? I think you're asking what grain of prediction, like spike by spike or average. So as I sort of alluded to, we're going to test average responses in IT over that 100 millisecond window. That was a very good predictor of the behavior from the earlier part of the talk that I showed you. We're not going to try to predict spike by spike. We're going to predict the mean response of each IT neuron to each image.
And where does that 100 millisecond delay coming to the model, is that at all?
The model does not have any temporal dynamics at all. This is like this large class of models that you probably already heard about. It's more about the spatial interaction across the visual field that we think these models are what's interesting about them. It's not a temporal model yet.
It's just that something you said earlier, I found very interesting, and I think that's missed in the model. The fact that when you go from station to station, it's just not simply a relay of information, but rather there's some computation in there. And so where is that in the model?
Good point, and I was sort of alluding to that when I say look at these arrows, right? There's local feedback, compute normalization, but there's no feedback in these models here. So any rumbling here would be independent of any feedback lines between areas. There's no cortical feedback models at all. And again, that makes, you could say it's missing something that biology has, but it's not, doesn't mean it's necessarily wrong. It was just a starting, we just don't have it there yet. So you might say, I think this gets to deep questions about, well, if you want to predict exact dynamics of B4, this model's not going to do that. It's not going to offer that well. And I would agree with you. We just wanted to predict, could we get the mean rates of IT over the images as a first test? That's all we've done. So you can test, you can push back on the grain of prediction. We're not going to predict every spike. We're not going to predict all the dynamics. But I tried to convince you earlier that it doesn't seem like we need much to predict those dynamics, at least up here, to actually explain the behavior. And that may not be fully satisfying for you, again, depends what your goal is, but that's what we've done. And we could imagine a dynamic model, we just don't have it yet. It's just already a hard problem of how you integrate across space and what nonlinearities do you use. And again, you might get my sense of my philosophy, start with something, even if it's sort of simple and wrong, but at least get you started and see how well you can do.
Do you guys know if the performance depends on the data set that you use?
The performance of the model, or?
I guess either way, for predicting.
I haven't even shown you if we could predict anything. Right.
But as you know, I sort of know.
Yeah, okay. So we can predict something, right? So, and I want to show everybody else that, but I think you're asking, how does the optimization target interact with the predictions? That's my translation. Or the model class interact with it?
Well, actually, in particular the data set, because, you constructed it to avoid correlation between background and others, but in the real world, there are correlations between both.
Right. Great. So maybe if we had actually optimized the model on those images, it would make better predictions.
Yeah, I'm just wondering if you guys have tried that.
Not yet. No. And you should talk to Dan in the lab if you want. He's interested in all of that stuff. And there's lots of cool things you could do in extension of this. And we just wanted to see, this is a different strategy. Just to point out, when people try to fit these neurons up here, they usually take the neural data and try to fit it directly. And that turns out to be there's not a lot of neural data, but you can get a lot of images out to test, to kind of optimize for. So this is a different way to get into the system to make a guess about what it's doing. And then post hoc after the predictor, not rather than fit the data directly. But that's what we need. The upshot is these models predict way better IT than previous models that we had in our hands, so we think that's powerful and useful and an interesting start. About 50% of the variance. Maybe I should show you that, because we're almost out of time. Aaron, you got your hand up. Maybe can I just show you guys the prediction, or?
Well, it relates to the last slide.
Okay.
What do we make of the number of parameters in the model?
Nothing other than it's a very complicated model, even though there's no feedback, which would be even more complicated.
So there are thousands of parameters?
Yeah. But they're locked down. Once we lock them, now we're not predicting neurons. Once we lock them, there's no more pre-parameters. So they're locked according to tasks, and then held, and I'm going to show you predictions of IT once parameters are locked. So no neural data are used to set those parameters.
So it's all about performance on the task?
That's right.
Okay.
And I shouldn't say no. For the aficionados, you might say, well, wait, how do you take a feature up here and map it to a single neuron? You have to take some data from the neuron there. What we do is linear regression from some of the IT data, each neuron, from the feature set here, linear regression, predict on the held out image response for the type. So there's a few parameters here, depending on how many neurons, or how many features that we have here that we have to regress on. So there's a little bit of data used to actually make the final regression. But all those other parameters there, all this stuff is all locked down. It's just the feature mapping here. We're sort of saying, it's just in the linear space spanned by the space and stuff like that. Okay, so here's the prediction. Okay, look, the black line is the prediction of the mean response in the 100 milliseconds of an IT neuron. Here's a bunch of images. It's not time, these are images. So you see some images are more or less in sort of category structure here. You can see for some reason that likes chairs. The red line is the prediction of this model. You can see it tracks really, really well. Its correlation, R squared is 0.48. That's like a correlation of 0.7. It's not perfect, but it's really, really good. And it's not just for, remember, these images, these are some example images here. These were never seen, the object images were never seen by this algorithm before. That's the sort of point we were just discussing. So to predict reasonably well over these kind of, not fully natural, but naturalistic space, which we measured IT neurons. Here's a sort of face neuron that, usually it's a face neuron, but there's always structure in these neurons. There's like some images of faces better than others. And look, there's a pattern here, a prediction that actually is followed even within this neuron. You might call simply a face neuron. It's not, the neuron doesn't respond to all faces, but that structure is also reasonably well captured. This is a different neuron. Here's another neuron that you wouldn't look at and categorize it easily as anything, but it can actually predict pretty well this neuron, something about the shape features, and it can predict, again, pretty well. These are all R-squareds, tend to be about 0.5, so about half the variance explained. I'll show you that in the context of other models and ideal observers as fraction explained variance. Take a bunch of other models and do exactly the same thing. Pixels V1-like, a V2-like model, H-max, another comp model that we built. A bunch of other models here, they all predicted about 0.2 on these data. This model, again, was really impressive that its top level was at 0.5. So again, it's a dramatic improvement, but not perfection, which would be 1.0. And each level gets better and better, so that's pretty cool. I think we gotta probably stop soon. Let me just tell you that we also, what's really neat, what Dan, the leader of this work, really likes is that we didn't just predict this, and there are no neural constraints, but like now you take this intermediate levels, where I showed you Ed Connor's work, it's very confusing to know what words should we use. Sure, people like the word faces and semantic words, and they think they understand. Before, these intermediaries are very mysterious, but you can take these models that have been optimized for these object tasks, and then you can look at their intermediate levels, and they like the intermediate levels of this model, for some reason, L3 predicts the V4 responses really, really well, even though, again, never saw V4 neurons. It's better than all these other models here. The intermediate level, and the top layer predicts it worse. So it's really striking how you just optimize these class of models, and you can already predict not the high layer, but also the intermediate level, at least at the grain that we've been measuring, and 100 millisecond mean responses for each image. I'm just going to close this, put this up. This is a Chris Gorte. This is a population level metric. By these kind of metrics, we're almost perfect, which is, this is a more compressed grain of analysis here. We're almost perfect at predicting these kind of Chris Gorte indices. Those of you who know that stuff. If you don't worry about it. I just want to leave this group with this kind of take-home message of what we did, and maybe you guys can discuss in context of CVMM. This is the performance of an algorithm on a high brain recognition, and this is why we did this whole thing. I should show you this in the beginning. This is the ability of a model to predict IQ responses. These dots are samples out of that model class I showed you. These are different parameter sets within that large family of models. Here's some existing models from previously. There's HMAX. Here's a model we built, Fossil 9. There's a V2-like model, V1-like SIF, Pixels. What I want you to see is that there's structure here, that the better you do on this, the better you do on that. And so this, again, supports the idea that if you can optimize on this, then you might do better on that, which is one of the things neuroscientists want to explain. So what we did was optimize here. We found a model that I call HMO, and it's now up to 50%. And the obvious thing to do is, well, if you can keep optimizing through the kind of right path with the right model space, you should be able to better and better predict. And that's kind of what we're trying to do next. So that's the spirit of what we did. We can get encoding functions out and talk about evolution development. And I'll just end by saying that kind of what needs to be done next. We need to start predicting for each image. We already mentioned this. We need to talk about dynamics and feedback, which has come up in questions about these kind of algorithms. We need to test the direct neural perturbations. I alluded to, we have proof of principle, but we have many more tests to do. We have to make these encoding algorithms. They're just a starting point. I gave you one called HMO. That's just one example in this family. How do we fit the rest of the variants in those encoding algorithms? That's a good discussion to have. Can they be unsupervised or more developmentally appropriate? To Emily's question earlier, these are all open questions, and that's sort of for the field and you guys to chew on. I'm telling you kind of what we've done. And I think this came up early in the discussion. This is one set of visual tasks that I call core recognition 1.0. We're thinking about 2.0. We're working on 2.0. What should those tasks be? We want to expand this domain of tasks here so that the grow these circles can be bigger and bigger, so to speak. And of course, we need to do that as well. But I would message to all of you guys is, to me, as an experimental scientist, algorithms that predict stuff, good. The more that we have, the better. We can test them against experimental data. You know, that just building models is a useful thing to do because that helps us to advance our field. These guys did all the work. I thanked them along the way. Ha-Ha and Dan Yeamans especially for the work I showed you through most of this talk here. And the folks who ran it as well. Funding agencies, and thank you guys for listening to my talk. So that's so Tom, you have to cut us off. I'm sure there's I took up all the time here.
No, we have 5 minutes.
Okay.
So one maybe three takeaway from the modeling work is that the IT responses didn't constrain your model at all. They're just used to validate it. So I do think we're just not at the resolution or the scale where we can study the brain to inform models, or?
I think, yeah, when we do this work, that makes me think, I often in the lab find myself thinking the exact same thing, like, what's the value of the neuroscience to the modeling? And, when we started, so I would say, because if you look at neuroscience, most neuroscience is actually trying to confirm things that engineers know how to do already. So they're taking simple models of decision, and there's various models, and then you go to computational conferences where they're quantitatively applying things that engineers think are just easy to do, and they're just then figuring out where they live in the brain. I mean, it's a little oversimplified with that. But what was cool about this space of problems to me is that these are things that engineers didn't yet know how to do. That's how I motivated at the beginning of the talk. And the hope was that we could find ideas from the brain that would tell us what to do to help the engineers. But where it seems to be right now is that as the engineering is getting better, it's hard to tell whether these models, remember, they were inspired by things that neuroscientists did 30 years ago, so maybe like a long time, like now deep networks are all the rage, right? But those grew out of things that neuroscientists had constrained. So if there was inspiration there, eventually now it's producing things that you could use as just the referees on which one is correct, which is a little bit like the neuroscience I said earlier, but now in a more complicated domain. Even our work there is an example of that. Let's just try to be good engineers and then validate, as you said. And I think there's a good discussion to have, like what's the role of neuroscience in driving the next generation technology? Maybe it just has a very long time lag. I think this is what my colleagues might say, that it just doesn't, right immediately. It's the way we think about the brain and the things we learn does have impact on the kind of models that some of us, I hope some of you in the room, are building, as it did for Jeff Hinton and Jan and Tommy and others when they build those kind of classical models that now are having success. So, I mean, I hear you. We'd like to go in and sort of extract the secret magic and then hand it to the engineers. We still hope for that, but to be honest, it's really just the validation of an engineering model in a bioconstrained space by earlier neuroscience. That's one way to look at the work that I presented at the end. Sorry, you gave me a chance to spout on. Is that fair? Yeah, okay.
So could I just get a citation for the laws of RAD?
Sorry, the last talk?
The laws of RAD IT.
Oh, it's co-signed and it's under review. So.
Oh, okay.
So I can give you a co-sign abstract, but I don't know if they're still under review. But I'd be happy to send you a draft of it. Send me an email, yeah.
What do you mean by learning each image is not just resume?
Oh, predict each image. It came up in discussion. All we're doing is predicting the pattern of difficulty and the confusion pattern at the object level. We're not predicting image by image confusion yet, for instance. And we're not predicting the trial by trial confusion yet, which you have to do as monkey because you can't, got to issue the subject. So there's finer grained prediction. If you believe in an algorithm, it's better predict down to every grain in principle.
This isn't the behavior, but you do predict the disagreement activity.
Well, okay, so there's two predictions. This predicts behavior, that's what's here, those decoding algorithms. This predicts neural responses, again, at the grain of 100 millisecond mean responses. Okay? You might argue that that grain is not good enough, but to me, you take this to inspire what you should predict, and that's what we're trying to do.
But with more neurons, does it seem that it will go there?
This one or that one?
The predicting each image?
Predicting this one?
Just having more neurons.
Does the more neurons in the laws of RAD IT algorithm predict better? That was a question that came up. I said, my student, that's the same question I want him to work on, but I can tell you that it works for the measures we have up to about 150, and then you're already perfect. But now at the finer grains, it's not yet perfect, but can you tell, is there a trend that you're getting more perfect? I wish I could answer that for you. We have the data, we just haven't analyzed it yet. That way we haven't moved to those grains. We're still trying to publish the part of it, just mention on the other part. Yeah.
What do you think of as, do you have any guesses on core objects 2.0?
Oh, great question. So, occlusion is a big factor in computer vision that we didn't have at all. That's one of the things we want to do. We also want to think about tasks that are more not about necessarily identity. We can estimate, so I didn't show you this, but we can ask humans to estimate things like the position and pose of these objects. And it turns out that same algorithm predicts quite well the human abilities to estimate, at least out of IT, to estimate like the position of an object. You might say, well, V1 could do that. V1 can't do that on these kind of cluttered images, but it should bring these other latent variables. So
Position is at distance.
Position in the field, like here's an image, show me where was the object. I'll show you an image, where was the object? And those cluttered images, you can take IT and it can say, oh, it was over there, but you can't do that on V1 simulation. So I'm sorry, this was a little diversion, but part of 2.0 is not just expanding the image space, but expanding the goals of what things you're going to be able to predict. So it's not just category, but also some of the other related variables, which we can already try here. But what I was going to go to next is one of the postdocs in the lab wants to do things like estimating surface normals, for instance, and we estimate other kind of factors off the shapes that are not just about identity. So, but we have to get a whole bunch of behavioral data on that to measure probably more neural data. But our main thing for 2.0 is occlusion in multiple objects, just one or two. That's the thing that we think is missing here, and that we know is challenging for computer vision. If you have a suggestion, I'd love to hear it. We're also trying to mine ImageNet for interesting dynamics on like what recognition happens early and late. We came up with questions of feedback to say, can we enrich the set with things that tend to take a little longer? Like that says say a second longer, but 200 milliseconds long. So we find paths like that. These have to be challenging. We're hoping to find things of that type. They might not have names like occlusion, but they are deformable, but they might have image sets that we can pull down and say, let's enrich with those and see if we can see those emerging later in the responses of IT than earlier. That might imply some role of feedback. That's what we'd like to try to do as part of 2.0. Okay, sorry.
Yes, I think I missed it. So right now your prediction is not for each image, or
the prediction here
yes.
is for each image there, in that domain that I showed you, the domain that I generated.
Okay, so but your goal is to predict for each image. Sorry, the goal on the right.
This goal is to take a population pattern somewhere in the ventral stream and predict the human report.
Oh, okay.
Right, and so I showed you we could predict human reports from monkey data using an algorithm that I call log drag. So you can take population vectors here and predict this very, very well, although the discussion is not perfect yet.
Okay.
But that algorithm then makes predictions to the discussion about space neurons, for instance. Under that algorithm and an assumption about spatial layout, you can lesion parts of this and make predictions of what behavior should be affected.
Okay.
So that's why this model has value. That's what I was talking about the object genetics for. That's, to me, one of the reasons it has value. It also has value because it demystifies, and maybe this was the earlier discussion, demystifies for us that you don't need to do anything complex to get to that. And maybe that pushes us to harder tasks or harder reports. That was a discussion you and I were having earlier. But that's sort of the value of doing these things. And I just want to point that out, that's something that visual neuroscientists spend most of their time over here. This is the history of visual neuroscience, Bill, Hubel, and Wiesel. So they've done a little bit of this, but they've neglected this for a long time, spending most of their time trying to build good encoding algorithms. And that's not bad work. It's just, we think that this really helps that as well. It tells you what you need to predict. And it's also deeply interesting in its own right? You push neurons around and change perception. That's DMI stuff, right? That's pretty cool, right? That's the deepest question that neuroscience are after. So again, I'm trying to leave you guys with the idea that just predictive models are good, and this is how we do them, and you guys are being trained to be able to build these kind of things, and that's the field, at least field of neuroscience, needs more of. So I hope that you will be attracted to this kind of work. And part of what my lab should do as an experimental lab is help to ask which models are correct and which are not, and how are they incorrect, and that's what we all need to do together.
So going back to the idea of the object manifold, to what extent do you think they are related to motor degrees of freedom, or sort of how we interact with the environment?
Yeah, that's a great question. That's sort of related to the 2.0 question. We're in this sort of limited domain of not vision, not just visual object recognition, core recognition. You're asking as we sort of expand the domain for gets to time, like now you're going to reach to an object, you're going to interact with it.
No, just move your sensory system around and see how.
Yeah, so now we've got time-varying movies is what you have to be showing the visual system to emulate that. And again, sort of part of our experimental necessity, not necessity, but constraint, it was simpler to stay in this domain of a 200 millisecond or 100 millisecond snapshot. Time-varying images will be really interesting, but what you'd like to motivate from is an interesting behavioral phenomenon that evolves over time. You'd like to show, like Leda and Tommy are doing, like action recognition, things that often require time. You'd like to have that It's not just start, as an experiment, you can't just start showing movies to monkeys. It's costly to do these experiments. They take a long time. So if you get motivated with this particular behavior up here, that's the way into that kind of question. That's why I would put that object recognition 2.0 or 3.0, or call it object interaction 2.0, whatever behavior you want. But if I could leave you guys with motivation, it's like define it from the top, from a perceptual sense, a goal sense if you're an engineer, and then we could work into asking, well, is it supported here? Which part of the brain supports it? And what algorithms support it? That's just how I like to think about problems, rather than just dive into the middle of the visual system and later figure out, oh, there's some neurons and maybe they relate to the task. I'd rather define this domain behaviorally of the task we're interested in and try to sort of make it interesting as even a behavioral experiment before you go in and measure neurons. This just makes more sense to me from an experimental approach point of view. So if you have good ideas about that, I'd love to hear what you're thinking about and what we might be able to.
Actually, on that note, are there, do you have predictions about like what should happen if there's like a movie of an object undergoing some pretty straightforward transformation? Like, should you see?
Yeah, I mean, that's sort of getting into dynamics of the whole system question, right? I mean, you predict from these simple models, but you predict as soon as you show an object, it would, this model, take that face, you show that face neuron prediction, we had different views of the face, was sort of wobbling around. It would predict what that neuron should do over time. Now, in reality, the dynamics of the system, when you start to show a movie, are more complicated than the dynamics of stitching together static images like that, right? So, and I think, you guys are all pushing to, like, as you have time-based unfolding movies, how well are these kind of models going to work? And, I already know that they're not going to work. There's going to be dynamics models not going to capture. And so there really was meant to be a domain. Again, you define that a regional domain. You guys are rightly pushing me to go to a larger domain. We've done a little bit of work in that regard, but like take a neuron, I think it's a face neuron, right? Oh, so you put a face up, but you go, still responding, the face is still there, right? But in practice, it goes, okay, it's adapting, whatever word you want to use, but even if you don't move it around, you put the face up, it goes away. And you say, well, that's explaining away, the predictive coding, all of that could be part of that system. The neuroscience will just call it adaptation, that's not an explanation, it's just a description. And these models have none of that, and they have no predictive coding on them at all. That's one of the things that some of my postdocs want to do, is to say, could we layer on, now we have this core, could you layer on some of these ideas? And we're not the first to say that, but then could we do that and predict the dynamics, not just in a kind of explaining away, but put it in a larger theoretical framework of what should be